Instituto Nacional de Medicina Legal y Ciencias Forenses.
Hello, I'm Dr. Erick Bartelink from California State University, Chico, and I just want to thank the everyone who organized this conference for inviting me to speak today about the application of stable isotope forensics to human identification casework. So the title of my talk is Stable Isotope Forensics in the Provenancing of Unidentified Human Remains from Domestic and International Contexts. So I'm assuming most of you probably know what stable isotopes are, but just a very quick recap. They're basically atoms of the same element that have the same number of protons, but a different number of neutrons. And this just means that isotopes of a given element are going to go through chemical reactions at different rates based on their atomic mass. And typically when we sample human tissues for isotopes, we're looking at some kind of bulk dietary components of food and water that are consumed, as well as tracers to landscapes, such as places where food was grown. These are the isotopes that are most commonly used in forensic provenancing studies. So you'll see it includes light isotopes such as hydrogen, carbon, nitrogen, oxygen, sulfur, as well as a couple of heavy isotopes such as strontium and lead. And hydrogen, oxygen, strontium, and lead all provide information about origin, and carbon, nitrogen, and sulfur isotopes provide more information about diet, although there could be applications to origin with these isotope systems as well. There are a number of excellent books outlining applications of stable isotopes shown here. The ones on your right are specifically focused on forensics, including stable isotope forensics and food forensics. But these are just good resources if this is a field that you're interested in learning more about. So I'm going to start with carbon isotopes. And basically, you are what you eat plus a few per mil. And what that means is that different plants have different photosynthetic pathways, and there's a slight offset between the isotopic signature of the food and the tissue that incorporates that signature. And so plants that follow a C4 pathway, which are things like corn, millet, sorghum, amaranth, sugarcane, have more carbon-13 in them relative to C3 plants, which are things like wheat, rice, most grains, fruits, and vegetables. So these are the main two photosynthetic pathways. And what you're going to see is different isotope signatures incorporated into things like bones, teeth, hair, and finger and toenails, depending on the composition of the diet of the consumer. Just to showcase the differences between a C3 plant and C4 plant, you can see that they form a bimodal distribution. There is no overlap between these. So if you're a totally C3 consumer or C4 consumer, that would be really clear as measured in your tissues. If you're eating a combination of both C3 and C4, which most societies who eat C4 would be eating some of both, they're going to fall, the values will fall somewhere in between. And you can estimate approximately what percent of the diet is coming from C3 versus C4 pathways. For isotopes such as oxygen and hydrogen, these isotopes reflect hydrological patterns on a global scale. And so I have the United States shown here. The upper graph figure is oxygen isotopes, and the lower graph is hydrogen isotopes. And because oxygen and hydrogen form water, the patterns are very, very similar to one another. They practically show the same pattern. And you can see areas such as the Rocky Mountains, the area that's sort of in pink and purple color, are areas that have very low oxygen and hydrogen isotope values. So the further you are from the coast and the more you are in the mountains, the tendency is to have lower oxygen and hydrogen isotope values and higher values closer to the coast or other large bodies of water. And this reflects distance from the ocean. It's called a continentality effect, as well as elevation. And so you're going to get geospatial patterning in oxygen isotope waters. And these patterns can be looked at in precipitation water, but also more importantly, in tap water. And so, and they may not always align depending on whether precipitation water in a given region is also the same source that tap water is. The graph on the right is just showing you the meteoric water line, and it's just reflecting that global relationship between oxygen and hydrogen isotopes and how it varies between different climates. So warmer regions have higher isotope values for both isotopes, and towards the bottom left of the graph, you'll see more negative values in colder regions. Okay, so the idea is that you're going to incorporate these isotopes from water that you drink, as well as from food water, and it should reflect where you're getting your drinking water from. Obviously, much more complex than that in many different societies, but this is the general principle here. Strontium isotopes are a little bit different. It is a heavy isotope. It substitutes for calcium in the skeleton, including bones and teeth, as a trace element, and it can be measured in trace amounts with mass spectrometry. Strontium isotopes vary based on the age of the underlying geology, so the bedrock and soils. And strontium-87 is actually a decay product of strontium-87, and it makes it a radiogenic isotope, which just means it's a product of radioactive decay. You get lower ratios associated with younger geological areas and higher ratios associated with older areas. Again, this is showing you just the United States where I work, and showing you an isoscape, basically showing you the patterning in isotope ratios of strontium-87 86 in the continental United States. We can sample different teeth with stable isotopes, and they provide different information about the diet and origin of a person. In this case, we would be dealing with mostly deceased individuals who you cannot identify, and you're trying to use isotopes to try to narrow down a place of origin or a place of birth or a recent travel history. And it just depends on the tissue that you're sampling specifically. So if you're sampling teeth, for example, you're basically measuring the isotope signatures that are incorporated into the tooth signature at the time that tooth formed. And because teeth do not remodel, it will reflect the isotopic signatures at the time of formation. Whereas bone provides a much longer archive of time. And so if you're sampling from a femur, you could be actually sampling an interval of time on a scale of 10 to 20 years. If you're sampling a rib, you're probably sampling on a scale of probably like something like 5 to 10 years, because ribs remodel faster than a femoral midshaft. Okay, so we can get information about teeth from teeth and bones that will give us information about childhood and adulthood, origins and diet. Other tissues, if they're preserved and have not degraded, can be quite useful as well, such as hair samples. And each centimeter of a hair sample from a decedent reflects about one month of diet if you sample in 1 centimeter increments. And from fingernails, it's about 2 to 3 millimeters of fingernail sample reflects about a month of history. And if a person moves from one location to another, it takes around 30 days or about a month for their body water to turn over to the new source. And then that becomes the source pool where those hair and fingernail tissues are going to be drawing from. So there's a little bit of a lag effect when somebody migrates to a different location before it starts to show up in their tissues. So we have applied a lot of this to domestic casework in California. This is where I work. I'm in Northern California, about 90 miles north of Sacramento. And we do casework, forensic anthropology casework, for about 2/3 of the state of California. And this includes going to scenes with buried bodies, fire scenes, surface scattered remains, as well as analyzing remains for biological profile, for trauma analysis, and other identification methods. So one of the tools we have that we use for cold cases is to try to apply stable isotope analysis to unidentified remains when all other methods have not yielded an identification. So California has about 25% of all missing persons cases in the United States and about 20% of all unidentified remains cases. So we actually have the biggest problem in all of the United States with unidentified remains cases. So one case we applied this to was from our local county called Butte County, and the skeleton was recovered in 1998, a male between the ages of 28 and 42. We thought most likely Hispanic, but possibly Asian, based on craniometric analysis, and a stature of around 5'6", plus or minus about 4 inches. The individual was wearing clothing, and so it gives us some idea of an approximate time frame in which they were buried. They were wearing a 1984 Los Angeles Olympics T-shirt, so that means the maximum date of deposition would have been 1984, but of course it could have been after that. We found clothing and shoes. None of this information has helped to narrow down identity on this individual. This individual did not have any dental work. They had moderate dental wear and a number of linear enamel hypoplasia defects, which are not typically things that we see in the U.S. population. They're much more common in poorer nations like people who crossed the border from Guatemala, Oaxaca, Mexico, etc. We had evidence of trauma, blunt force trauma to the right side of the skull, suggesting that this was a probable homicide. And we did do isotope analysis, and we did carbon, nitrogen, oxygen, and strontium. And this isoscape prediction map basically shows areas where oxygen and strontium isotopes overlap from the same tooth sample. And so we had some places to the far left, just north of where the body was found in Northern California, some other locations in Southern California, and then some areas in the Southwest, such as New Mexico. So these are the possible locations, assuming the strontium and oxygen data are accurately predicting origin, where this person could have been from. However, the carbon isotope values were very different. We noticed that comparing to some food web data published by Nordotto and colleagues in 2006, that the bone collagen isotope signature was around -10 per mil. And that's an extremely high carbon isotope value. That's what we see actually in ancient Maya skeletons from Guatemala. And we think this information strongly suggests this person is likely from south of the border and may have been someone who worked in migrant farm work in Northern California. And the reason they haven't been identified could be because they were not reported missing. So we're doing additional work with this case. We'd like to do forensic genealogy to help advance the case along. DNA has been taken. The decedent's information is in the National Missing and Unidentified Persons System, and we've done everything possible so far, and we're hoping that forensic genealogy may help with identification of this individual, especially if they have relatives that live in the United States. Okay, another case we got was from San Bernardino County in 2010. We received this just a few years ago, though, for a reanalysis. And this was a female about 15 and a half to 19 and a half years of age, likely Hispanic, and of unknown height. The remains were found as a fleshed head that was located inside of a backpack in the desert in San Bernardino County. The decedent had likely only been dead a couple of days. Their skin of their face had been removed. It had been cut off. There are sharp first defects on the nasal bones, and the rest of the head was completely fleshed. And so we believe the person was decapitated and the skin of the face was removed to try to obscure identity. We used the London Atlas to provide a better age estimate than what had been previously provided. So we estimated between about 15 and a half and 19 and a half years. And this individual had a lot of very high quality dental work. So they had composite restorations in multiple teeth, suggesting that this was not someone who likely was homeless or transient. They also had a deviated nasal septum with a growth in the left side of the nasal aperture that's often referred to as a concha bullosa. So this is a potentially identifying characteristic of this individual. When we did our predictions, so San Bernardino County is shown over here on the left side and the very bottom in the small circle. And so that's where the body was recovered from. The areas with the larger red circles are possible places of origin based on oxygen and strontium isotope analysis of tooth enamel. And so the closest place would be in Arizona, so not too far from San Bernardino, just a few hours away. There's also a possibility in the northeastern United States, but that seems less likely. But technically, that would have to be included in the prediction. When we did bone, and we did not have enough funds to do strontium on bone, but we just did oxygen. It includes all the areas showed in blue, and we cannot exclude any of those areas. It actually does include the place where the head was found in the backpack. So it doesn't rule out that she may have been local to the area based on the bone signature. And then when we did hair, we did many different increments of the hair. So we serial sampled it. And when you looked at the last 13 to 3 months prior to death, it included a really wide area. And this just means the isotopic signature for oxygen isotopes could be found in many different locations, including the location where the head was found. So it doesn't answer the question of whether or not she lived there locally or not prior to her death. It includes many places in California, across the southwest of the U.S., and then into the Great Plains, into the Midwest and the Northeast. So not very useful, not very diagnostic. However, when we looked at the last 3 months prior to death, there was a significant shift to a much higher oxygen isotope value. And so there was very little variation from 13 to 3 months prior to death, but in the last 3 months prior to death, a very significant shift that suggested places like Texas, Oklahoma, and Florida, so along the Gulf Coast of the southern United States. And so we believe that in her last 3 months of life, she likely resided in one of these locations and ended up over on the west coast of California. It is along a major interstate, a major freeway that's used by truckers, people who drive semi-trucks. And her remains were found close to a truck stop. So that's one possible hypothesis that they're looking at. So we think it's possible that she may have been born in a place like Arizona, and then she may have resided in a place like Florida or Texas or Oklahoma prior to her death in the last 3 months. She is still unidentified, and they are doing forensic genealogy on this case. So we're hoping to get her identified. And we really hope that this information helps with narrowing down possible missing persons cases associated with her, because she is a young woman and would very likely have been reported missing by family and friends. Okay, now shifting gears here from away from domestic casework, dealing with cold cases, I'm now going to talk about larger scale applications to things like past wars and conflicts, as well as humanitarian efforts of identification. So one thing that we know about the United States is that sugar and corn form a fairly large contribution to the very processed diets that many Americans consume. And it has increased a lot over the last 50 to 70 years or so. So you would expect that, and we see this in isotope studies, that many US Americans have fairly high carbon isotope values, suggesting significant contributions of C4 resources to their diet, such as corn and corn products, as well as sugar. And previous work by Husselman and colleagues in 2015, and this is a very incomplete perspective based on a very limited sample, but it does show you there's some regional patterning in carbon isotopes globally. And so areas that are in red or orange have high contributions of C4 resources. Areas that are blue, more towards blue or purple, have very low C4 contributions to the diet. So you can see that there would be big differences between, let's say, the United States and perhaps Mexico and Central America, and big differences between, let's say, Southeast Asia and the United States. So we can use carbon isotopes to tell us some information about diet and also some information about origin in certain cases, depending on the specific hypothesis that you are testing. So I have been working with the Defense POW MIA Accounting Agency, DPAA, for many years on isotope-related research, and they have been interested in using carbon isotopes in particular to separate out the remains of local Southeast Asian individuals' remains versus U.S. American soldiers from the Vietnam War, as well as other conflicts in Korea and from the Korean War, and even contexts such as World War II. So we have wanted to understand whether or not carbon isotopes could separate out Southeast Asians versus U.S. Americans based on their dietary differences, and use this as a screening tool for which samples would be submitted to the DNA laboratory for identification. So when we did our first pilot study published in 2014 in Annals of Anthropological Practice, we found a bimodal distribution in both the collagen carbon isotopes as well as the bioapatite, the bone mineral carbon isotopes. The Asian-derived samples, which are primarily from Southeast Asia, really show very low carbon isotope values, indicating their diet is primarily C3 resources, whereas in the United States, we have a much stronger contribution from C4 plants. And so you can see they form two different distributions, whether you're looking at collagen or bioapatite. And statistically, we could say that there was a greater than a 95% correct classification rate in assigning a individual to either Southeast Asian origin or U.S. American origin, just based on a simple linear discriminant function analysis with cross-validation. So this is a powerful tool. The DPAA laboratory has expanded on this significantly since the publication of this paper, and they actually have an online free app called Isolocate, where you can actually plug in isotope values, select certain samples, and try to predict origin based on carbon isotopes as well as other isotope systems. It's obviously still very preliminary, and you have to have a really well-constrained hypothesis that you're testing, such as are these remains from Southeast Asia or the United States? We have published a number of papers. These are collaborative efforts involving identifying U.S. service personnel from past wars and conflicts, as well as doing studies on intraperson variation in isotope values. So a recent paper we published, we actually looked at how much variation is there between skeletal elements of a single skeleton. That was published in Forensic Science International recently. And then we also looked at some methods for improving quality control of bioapatite for stable isotope analysis. And when do you not use the values based on evidence of chemical alteration or diagenesis? Okay, now I'm going to switch gears here to a third project, and this is one involving identifying or using stable isotopes to try to identify human remains from the U.S.-Mexico border of deceased migrants from Mexico and Central America primarily. Some of this work has been published in the Wiley Forensic Science and Focus series. So there are these two excellent volumes edited by Roberto Parra, Sarah Zapico, and Douglas Ubelacher that I would strongly recommend. There are many stable isotope chapters in these books, and they're an excellent set of resources. So I'm going to be specifically talking about the U.S.-Mexico border, and you can see, I'm sure you know, it's very militarized. We have extensive border fencing that's been put up over the last few decades in order to block undocumented migrants from crossing. And of course, the result of this has been to drive migrants through much more dangerous environmental circumstances. So, for example, they're trying to pass through the Sonora deserts to try to get into the Tucson area of Arizona. And we know that every year, hundreds of migrants are dying due to dehydration and exposure going through these very dangerous areas. So, and in Texas, we're seeing a similar situation, but often migrants are making their way several miles into Texas, and they're trying to walk around a border checkpoint. And many of them die as they become lost and do not have enough water. And it's very flat landscape, so it's very difficult to see which direction you're walking. So these are areas where you see large quantities of deaths. We know that migrants take different routes into the United States, and these could have a bearing on where they come from. So some routes tend to be used more by Central Americans and others by Mexican migrants. And we know that a lot of them are taking very dangerous paths towards Mexico and into the United States, such as riding the Beast here, as it's often known. A lot of people get injured and falling off of trains, get injured, or get assaulted or robbed of their funds for traveling as well. So I'm sure you're very familiar with these issues, but very dire humanitarian circumstances. So in Texas, in particular, individuals have made it into the United States. I think they're over 40 miles north of the border, and they're trying to bypass this Falfurrias checkpoint, which is a small town in Brooks County, Texas. And basically, individuals who get lost, injured, or are suffering from dehydration or sunstroke, heat stroke, are often dying on private ranches. And so they're not often found for many weeks to months, sometimes years, or not at all. And so there's been a strong effort by universities such as Texas State University, Baylor University in Texas, and University of Indianapolis, and also a number of non-government organizations to try to not only find bodies and get them identified and recovered, but also to try to provide things like water stations for migrants in these areas. So it's a very serious humanitarian issue, and it's one that the United States government has not paid a whole lot of attention to, unfortunately. So we know that, like, Dr. Kate Spradley has led the Operation Identification at Texas State, and she has done wonderful work in creating identification processes for deceased migrants from South Texas. And this is just a very incomplete map, just showing you deaths around that Falfurrias checkpoint. And these are just between 2011 and 2013. And this is an incomplete map, but you can see that this is essentially just a massive number of fatalities around this very small town. And it's completely caused by people trying to bypass this checkpoint. Okay, this is just another view there of the concentration of deaths in Brooks County. Okay, most of the deceased migrants in the past were buried as unknowns, unidentified remains, in a cemetery in Falfurrias called Sacred Heart Cemetery. And so exhumations occurred and have occurred over a number of years by Texas State University, University of Indianapolis, and also Baylor University. And as individuals have gotten identified, they have noticed that there's a high number of Central American migrants that are dying in South Texas, a much higher number than found in Arizona in the Tucson area. So Tucson gets more Mexican nationals, and South Texas gets more Central American nationals. And again, it just reflects the nature of the migration routes and where people are traveling from and to. So these are important things to know in terms of identification, as well as where are people coming from. And of course, we can only know that once they're identified. So Operation Identification, run by Texas State University and other collaborators, they're interested in things like the biological profile, obtaining DNA samples, recording information from dental work, and also personal effects. And stable isotope analysis has been a new thing that has been applied to assist with this issue in the last 6 or 7 years. And so I've been fortunate enough to work with Texas State University and Pima County in Arizona to sample remains of deceased migrants and to try to generate methods to predict region of origin. And I have many collaborators who have done a lot of work on this issue, like Dr. Saskia Ammer, Dr. Robin Kramer, as well as some of my former graduate students, and a recent grad student, Taylor Lamberger, has been doing research on collecting water samples from Oaxaca. That's been very interesting. Okay, so how could stable isotope analysis aid in the identification of undocumented border crossers? We have to think of this as not identification so much, but as narrowing a region of origin. And we need to understand how much isotope values vary within Latin America in order to get a sense of how well we can apply these methods. We need to know about variation in diet, water source, and geology in the areas where migrants are coming from. So how can we use this to sort of narrow down a possible region of origin? And what are some of the limitations and challenges of these methods? There are many, and my feeling is that we're in just very early stages of this kind of research. So the use of multiple isotopes is definitely a good thing for helping narrow down region of origin, because the more isotopes you use, the more that you can identify specific differences between groups. So we have found that carbon isotopes can be used to differentiate between US Americans and undocumented border crossers in most cases. Much like we found that carbon isotopes could differentiate between Southeast Asian individuals and US Americans. And this just reflects differences in C4 versus C3 food consumption. You'll see shortly that undocumented border crossers have much higher carbon isotope values than most US Americans. Once you've done that first step using carbon as a potential screening tool, then you can use oxygen isotopes to maybe at least suggest whether somebody's closer to the coast or more inland within a region within Latin America. Ideally, we want to narrow it down even further. So things like strontium isotopes, I think eventually will have a stronger role to play than it currently does. We don't have a lot of baseline data for strontium in Mexico and Central America, unfortunately. We are just starting to get more data on oxygen isotopes. You can use discriminant function analysis or decision tree methods to work through the process with different isotope systems. So you can use carbon to sort of say, okay, are they likely a migrant or not? And then use another series of isotopes to try to narrow down origin even further. So we have a lot of assumptions. We assume that human diet is patterned by things such as ethnicity and region, as reflected in carbon and also nitrogen isotopes. It does turn out that carbon is much more useful than nitrogen. Nitrogen values don't vary that much between the groups that we have looked at so far. So it does help in a small way, but carbon is much more powerful. And we are assuming that most food that is consumed in the areas where these migrants are coming from is locally grown, as reflected in strontium isotope ratios. We know that, like in places like the United States, food can be imported from far away, and that it can be very difficult to say that the strontium value in a person's bone or tooth is reflective of the local geology. So that's something that we have to do a lot more research on in the United States. However, we're assuming that many of these migrants that are crossing, they tend to be more indigenous Maya. They tend to eat more locally grown foods, And we're hoping that it's a safe assumption that the strontium values in their tissues do reflect the local geological signature. Again, more research needs to be done on this because these assumptions may not be correct or may only be partially correct in certain circumstances. We also are assuming that most people are drinking or imbibing water that is local. And that's a safe assumption in many places, but as my former graduate student Taylor Lamberger found in Oaxaca, is that a lot of people were getting water from bottled water or getting water from water trucks. And so it wasn't necessarily local in many instances. And so we really have to do a lot more research on the sources of drinking water that migrants are using before they cross, so that we have a better understanding of how to apply these methods for predicting origin. Okay, so again, these are assumptions that we're making, but they may not be correct. If you drink, for example, very expensive Fiji water, as you can see in the lower photo, it actually is from Fiji. So if you just drink that and that alone, your hair values over time would start to look like Fiji water. Okay. So it's best to use isotopes to test specific hypotheses, so as a possible exclusionary tool to narrow down possibilities. So maybe identifying whether a person is likely local or non-local to the area where they died, or if the person might be non-local to the country where they died. So these are going to be the most sort of powerful kinds of applications is when the signatures of the tissues look very different from what local individuals look like. So it has to be contextualized with your specific study. So applying this to undocumented border crossers, we have found that individuals that are from Central America, basically from Central America and Mexico, have very strong or very high carbon isotope values, much higher than the United States. And so this is a strong contribution primarily from corn or maize products, and also some from sugar. In the United States, its carbon isotope values are very high, but not nearly as high as in the migrant groups. If we look at a place like Korea, they're even lower than the United States in terms of carbon isotopes. And they do consume some millet and corn in Korea, and that's why they have some C4 signature. And then if we go over to Southeast Asia, where dominant staples include C3 resources such as rice, you'll see that the values are very, very low, indicating almost no C4 contribution to the diet. So this is spatial patterning on a very gross scale, but it can be useful for making decisions about probable origin of individuals. We can't assume every individual that dies in the desert in Tucson or South Texas is automatically a migrant just based on discovery of the body in those locations. We need to do other tests to help support or refute that specific hypothesis. So like we did with the DPAA study, when we looked at undocumented border crossers versus US Americans, we could correctly classify individuals in terms of their origin about 96% of the time, just using a simple discriminant function analysis. Again, this is still kind of preliminary, but at least statistically, we can say that there's some pretty big differences here. We also can see that there are groups among the migrant sample of undocumented border crossers that fall into a lower C4 group, a higher C4 group, and kind of an in-between medium group. So there's almost like a multimodal distribution here, suggesting that there's different groupings of migrants that have different amounts of C4 or likely corn contribution to their diet. So when we look at these groups, they do sort out pretty nicely based on comparing bone collagen groupings for low, medium, and high C4 diet versus their tooth enamel values, which would be tracking their childhood diet. And we can see pretty strong correspondence between childhood diet and adult diet. So for example, the correlation between an individual's tooth enamel carbon isotope signature versus their adult bone signature is 0.804 for an R square value, which is a very strong correlation. And so the amount of C4 in the diet seems to be maintained between childhood and adulthood for the majority of the individuals that we have examined so far. Okay, so this suggests that individuals are eating the same kinds of foods as adults as they were as children in terms of their isotopic composition. We do need a lot more reference data for Mexico and Central American countries so that we can develop higher resolution isoscape maps. And one has been completed recently by Dr. Saskia Armour as part of her dissertation work, which was published in 2001 in the Journal of Forensic Sciences. And so she was able to collect data on water samples throughout Mexico. And you can see there's strong geospatial patterning here in both oxygen and hydrogen isotopes, much like the maps you saw for the United States. This is still a crude map. We need more data points. Dr. Amr also collected hair samples, and so she has also looked at the correlation between hair collected from hair salons and isotopic signatures in the tap water from local communities. So it's a good start. We need a lot more data, and we need a lot more nuanced data, specifically to address the fact that a lot of individuals are getting water from different sources like water trucks, bottled water, especially if the local tap water is too contaminated to drink. We're also seeing some patterning with strontium isotopes. So we've looked at strontium isotopes in bone and also in tooth enamel and have found some differences there. There's a wider range of strontium values in the tooth enamel than there is in the bone, and that could be for a number of reasons. Perhaps the childhood origins are more diverse locations than the adult origins, but it all pretty much falls nicely within the kind of areas of Central America that we would expect for many of these individuals based on previous bioarchaeological research conducted by Dr. Lori Wright and others. We can also see there's some patterning between strontium isotopes and diet. And so you can see some clusterings of the individuals who have a C4 diet that's low C4 diet, a medium C4 diet, and a high C4 diet. So I do feel like we're pulling out different groupings of migrants, likely from different areas and in these different dietary categories. At this point, it's just simply an observation of a pattern, but we need to do more research. And one of the expectations is that the individuals with the highest C4 diet are going to be likely the most indigenous populations, the ones that are going to be, mostly indigenous Maya who eat more C4 products. A couple of case studies where we've done predictions. So we have one that was done with oxygen isotopes only, and it predicts some areas within the United States, obviously several within Mexico, but it was consistent with the origin in Guatemala where this person was from. But you can see there's a lot of areas that are consistent, so not super useful in terms of really narrowing it down that much. But again, this is sort of an early start here. We do have a strontium signature value that was consistent with that area as well. Another case we did, or this is actually the same case, you can see the areas that are consistent with the strontium value for this individual, although we don't have great baseline data at this point. Okay, and then this is a recent study by Laffoon, also mapping out some areas of Central America as well, also showing it's consistent with that signature. Another case that we did was very interesting because the areas predicted by oxygen and by strontium, as well as where they overlap, which are the areas in red, were only consistent with the United States and not with anywhere in Mexico or Central America. So when we also look at this person's dietary signature, they are below the box and whisker plot for the undocumented border crosser sample, suggesting they're a potential outlier or atypical case. Their isotope signature looks more like U.S. Americans than it does like the undocumented border crossers. So there are several possibilities for this individual. One is that they're not a border crosser. Another possibility is there's someone who maybe grew up in the United States, but maybe was deported and died trying to cross back into the United States, and that they have a U.S. signature because they were living in the United States, but perhaps got deported. We do know of some cases of that happening. And there are other possibilities that they could be a migrant that's not from the countries that we have sampled so far, so perhaps from somewhere else. We are going to look at some of these cases using forensic genealogy and see if we can get any more headway on the identification of those individuals. Okay, so I'm going to wrap up with some limitations and challenges. We obviously know that food being imported not only within countries but between countries can create this global supermarket phenomenon where people are eating a lot of non-local foods. We also know that bottled water may often not reflect the local drinking water, although many times bottled water is purified local tap water from some communities. So a lot more work has to be done to explore that issue even further, and how that might affect stable isotope predictions. We need to have better maps based on tap water so that we can compare that with precipitation water and make decisions on which maps are the most useful for making predictions in a specific area. So we definitely need a lot more data into isoscapes. And we need more reference data so that we can do better interpolations of areas that we don't have data for. And we also have to acknowledge that how we prepare our samples and how we run them on mass spectrometers can actually affect the results. This is a study by Pestel and colleagues from 2014, published in PLOS One. And you can see there's 20 different laboratories that analyzed the same femur sample. So it's all the same bone. And each lab analyzed 5 pieces of bone from that femur. And you can see how much variation there is in the oxygen isotope values based on the laboratory where the sample was run. And so what this means is that different labs are preparing bone bioappetite samples differently, and also running them using different settings, like differences in acidification temperature. And this is creating a lot of noise and a lot of variation. And so if we're not getting the right value for the oxygen isotopes, we're going to make incorrect predictions of origin. So this is an area where a lot more work has to be done. More work needs to be done on teeth as well. But it's just something I wanted to kind of end with, just to show that isotope values can vary depending on how samples are prepared and where you run those samples. I'd like to acknowledge a lot of this research was funded through the AAFS Humanitarian and Human Rights Resource Center, as well as Atlantis Foundation Fellowship through California State University, Chico. I've had a number of collaborators who have assisted and collaborated on these projects that I'd like to acknowledge as well. And that is the end of my presentation. Thank you very much.
Instituto Nacional de Medicina Legal y Ciencias Forenses.